Our last talk of today is Linda Kinkel, and I am really proud to introduce her today. She is Professor of Plant Pathology at the University of Minnesota. She completed graduate studies at the University of Wisconsin-Madison and postdoctoral research just down there somewhere, right? Yep, just down a hill, great. And her research focuses on ecology and evolutionary biology of plant-associated microbes in native and agricultural habitats. And I guess that's what you're going to talk about today.
I realize I stand between you and the poster session, which presumably includes some snacks. Am I right? Okay. Okay, so I'll try to be efficient, but I have to say the talks today have been great. I love coming and seeing the diversity and the way that the talks have been mixed up today, that they're not all technical, that they're conceptual and tools and ideas and new models and platforms. Just a fabulous day. So I'm really grateful to be here and glad to be here with you today. I'm preaching to the choir to say that plant and soil microbiomes are critical to plant health and productivity. We know this. And we, by and large, all want to lean into this. How do we manage these communities effectively and actively to achieve the desired functions, be it disease suppression, P-sol, Z-sol, plant growth promotion, drought resilience? How can we manage? And this really is two distinct questions. And I think there's two questions that we need to ask to really get to the management outcome that we all, I think, hope for. First is if we want to manage to achieve function, we have to understand the role of that function to the microbe, right? What is the role of this thing that they're doing to the fitness of the microbe itself? And the second thing we need to understand is what factors mediate selection for that function in the natural habitat, which has to do, again, with what does that function do to confer fitness to the microbe? And second of all, what factors influence the expression of that function? We've heard a lot about signaling today and homoserine lactones. What factors determine whether or not that expression or that gene or that biosynthetic pathway is expressed? Now, today I want to talk about plant disease suppression. Our work is focused predominantly on streptomyces within soils. I started working on naturally occurring disease suppressive soil that had very high densities of inhibitory streptomyces. And we long ago, and we still are continuing to work with microbial inoculants, and I actually think this doesn't work, which is totally fine. We showed that we could inoculate. On the left, we can inoculate soils. GS93 is one of our great antibiotic producers. It suppresses Phytophthora alfalfa as compared with the control. These are naturally infested soils. Similarly, for potato scab, when we inoculated on the left versus non-inoculated, we did a great job at suppressing plant pathogens. But short of inoculation or beyond inoculation, what is it that we can do to create naturally occurring disease suppressive potential, right? Every soil has streptomyces and antibiotic producing microbes. How can we enrich these populations or turn on their functions to effectively achieve this beneficial outcome? Now, to answer this question, as I said, we need to understand what is the role of this beneficial function for us? What is the role of antibiotics in the ecology and evolution of these populations in soil? Now, the short answer is an easy one that we all learn in introductory microbiology. Well, antibiotics are weapons, right? And in fact, I went to two meetings in particular. Julian Davies, who many of you might be aware of his work, fought back really hard. He said, no, antibiotics are not weapons. Because antibiotics also at very low concentrations mediate gene expression. They change expression of many biosynthetic pathways and genes throughout the genome. And so we realized that we actually needed to know, are there weapons? Are they weapons? Because this influences how you might impose selection for this function. So we decided that if antibiotics were weapons, in fact, naturally occurring communities should be able to tell us. And in particular, if antibiotics are weapons, we should see evidence in sympatric populations that are together in soil. We're looking in soil here. Sympatric populations should show evidence for selection for inhibitory capacities. That is, I should be better at killing things that I live with than things that I've never seen before, if antibiotics are effectively weapons that enhance your fitness. And so we asked this question, and in fact, I went around the world. We actually have collections from all the continents, and we collected streptomyces, and we looked at the extent to which isolates could inhibit and how well they could inhibit sympatric isolates versus allopatric isolates. So in the top figure, we have inhibition zone size on the y-axis, and sympatric, I'm a little worried about using that. The blue bar is sympatric strain pairs. This is a total of 90 isolates. So it's 8,100 combinations of isolates. The mean inhibition zone size among sympatric isolates in blue, among allopatric and yellow, we could see that isolates are in aggregate significantly better at killing things they live with than things that they don't. And when we looked at individual locations, this includes locations from Minnesota and Panama and France and New Zealand, and we found that at every location, within locations, we found that sympatric inhibition was greater than allopatric inhibition. So broadly, we found that there was support for this idea that there's selection for antibiotics as weapons to inhibit things that you live with. But it was a little bit more complicated than that, or I should say more interesting at the same time. When you think about what's the purpose of inhibiting populations, it usually, we think of competition and what do you compete for? Space and food. And so among these isolates, we also did a biolog SFP2 plate, which measures growth on each of 95 different carbon substrates. And we looked at the relationship between niche overlap between pairs of isolates and how well or how strongly isolates inhibited one another. How good are you at killing isolates? And we found that isolates are significantly better at killing sympatric isolates that have high niche overlap. So I have the relationship here, the bright blue, that's for sympatric populations or sympatric strain pairs. Among allopatric strain pairs, there's no relationship between niche overlap and how good you are or how effectively you inhibit another isolate, indicating that this isn't a genomic or physiological constraint. It strengthens the suggestion that there really is selection to inhibit sympatric competitors. So thinking of management, so great if they inhibit sympatric competitors, how can we lean into this? So this relationship between niche overlap and inhibition actually shows the risk of leaning into this. That is, if you look at the top panel on the right, you've got species 1 and species 2, two species that have a strongly overlapping niche. Now, this actually suggests there's two possible options. One, if I'm species 1 and you're species 2, I can kill you, then I win. But the other option is killing you. Antibiotics are metabolically expensive. What if we differentiate among our niches? What if, again, I use the cookie story. Some of you have probably heard this before. There's Nutter Butters and Oreos. I'll eat the Nutter Butters. You eat the Oreos. We might mutually optimize our fitness, not by investing in secondary metabolites. And so that work was on streptomyces. I want to note that with JGI, in addition to streptomyces pairs, and I'll get to our project soon, we've also done some work on cross-kingdom inhibitory interactions. And these are streptomyces and the fungus fusarium. And just to go really quickly through this, it's a newer project that we have. On the left, if you look at the x-axis, we have saprophytic fusarium and streptomyces from native prairies that are sympatric in the black dots. Each dot is one fusarium streptomyces strain pair. The x-axis is how much niche overlap does a fusarium have against an individual streptomyces? What's the threat, the competitive threat of a fusarium against the streptomyces? And the y-axis, again, in the left panel, we have how well does the streptomyces inhibit that fusarium? And you can see among sympatric, again, the bigger the competitive threat, the better the streptomyces is at inhibiting the fusarium. But we see no relationship, red, among the allopatric isolates. And these are isolates in soil locations that are about 20 meters apart. So you see this is very local coevolutionary dynamics. On the right, we have the reciprocal. That is, if the streptomyces has strong niche overlap with fusarium, how well does the fusarium kill the streptomyces? Again, with the black line is sympatric. We see a strong responsiveness of fusarium's capacity to kill streptomyces that represent a competitive threat and that live in the same place. And so really for across kingdoms within the streptomyces, in both cases, we came up with a way of thinking where I said, okay, so species interactions, microbial species interactions, this isn't a surprise to anyone in this room. They're really important for generating functionality. But in this case, we're talking about selective enrichment of particular functions. And our data suggests that these interactions might generate one of two distinct coevolutionary trajectories. First, we might have a traditional arms race, right? Antibiotics, you selectively enrich antibiotics, that imposes selection pressure on target audiences to reciprocate. And this generates what we believe this is the pathway that leads to these highly pathogen suppressive, naturally occurring soil communities. Honestly, we think the pathogen is irrelevant to this development. It's all about the saprophytes. The pathogens are just collateral damage. But of course, we noted that niche differentiation offers an alternative, right? Again, if there's a whole lot of different kinds of cookies, I eat the Oreos, you eat the Nutter Butters, we don't have to produce these costly secondary metabolites. And we might end up, in fact, with a very resource cycling efficient community. Niche differentiated communities tend to be very efficient on targeted microbes or targeted carbon substrates in cycling them, but not good at pathogen suppression. So this suggests that these are levers for selection, which is to say levers for management, which raised the question of what are the drivers? And we knew going into this that in our prairie systems, we were working in experimental prairies that were about 20 years old. The monoculture prairies were substantially more antagonistic than the polyculture prairies. So we thought about it, and this is where the cookies come into play. If you have a monoculture, we reasoned, well, gosh, isn't that kind of like having just one or a couple kinds of cookies versus a polyculture with lots of different cookies. And so we asked in these antagonistic monocultures versus the polycultures, is there a difference in the diversity of carbon substrates of microbial food in the soil? And I only want to spend a moment on this because I want to go back to antibiotics, but we did show we have Andropogon girardii, which is big blue stem, and Lespedeza capitata bush clover. And we looked at carbon compound richness using pyrolysis GC mass spec in the soil when the soil was sampled from the rhizosphere of Andropogon when the Andropogon was growing in monoculture in red, polyculture in blue, and the same thing for Lespedeza. And we did find that carbon richness was dramatically different in the rhizosphere when you have plants growing in monoculture versus polyculture. We followed this up by actually feeding soil mesocosms with one type of carbon or multiple carbons over 9-month periods. And we've shown and collected isolate, developed isolate collections. We've shown that we can have significant shifts in both antagonistic activity and nutrient use phenotypes in as short as 9 months under different carbon selection, which raises the question, why aren't we thinking about microbial nutrition the way we thought about plant nutrition? How do we feed microbiomes to achieve these functions? I think there's a lot we can do there, but let's lean back into antibiotics. So antibiotics, there's evidence that antibiotics are weapons. But we also know that antibiotics at sub-inhibitory concentrations can do lots of things that might not be about being weapons. And because I was challenged at a talk to say, and I was told that I was just using a human analogy to think of antibiotics as weapons. So I went back to the lab and I thought, okay, I can answer this question. If antibiotics are not weapons, maybe they are common in soil at sub-inhibitory concentrations. Maybe they rarely achieve those inhibitory phenotypes we see in the lab. But then we still have a testable hypothesis. If antibiotics are mostly present as signals at sub-inhibitory concentrations, how is that likely to mediate the way that the microbes interact? And so what we started with, and we did this about 10 years ago, it's a fairly simple study that we've been building on pretty aggressively in the last couple of years. We said, this isn't a hard thing to do. We took 9 of our streptomyces. We chose them because they were phylogenetically diverse. We took 5 antibiotics that all are produced by streptomyces. We wanted it to have at least a little ecological and evolutionary relevance, even though it was a pure lab study. We calculated the minimum inhibitory concentration for each isolate, and the SICA, or the sub-inhibitory concentration, that's formally defined as 10% of the MIC. So we went with what the literature told us to do, and then what we did is we characterized the growth of every one of these 9 isolates in a biolog plate without any SICA, and then with each of the 5 SICAs. So they're all growing alone in a biolog plate, with or without a SICA. And then we said, what do SICAs do to growth? And you can look at the paper if you want to look at the details. There was a lot of variability in how isolates responded to SICA. Many showed some decreases in niche width. A couple showed increases. I was really surprised to find out that some of these streptomyces grew on metabolites, grew on carbon substrates in the presence of SICA that they didn't grow on when the SICA wasn't present. So isolates varied and antibiotics varied. But of course, at the end of the day, that's just phenotype. That's just a phenomenon. What does it have to do with how microbes interact? And so we talked about niche overlap before, but I want to rethink a little bit how we think about the core question, which is, can a SICA influence the way species interact? And in particular, could a SICA confer a benefit? So if you think about the red circle is the total niche, like all of the nutrients I can grow on it and how much I can grow on it. And the blue circle in the center is a different microbe. Again, it's total niche. We can look at a niche overlap as where those 2 are confluent, right? The purple. But what we wanted to focus on here is the part that's not niche overlap. That is, if you and I are competing and we have 20% niche overlap, or I have 20%, Emily has 20% niche overlap of me, my red area is 80%. And it's actually an absolute number that we have, the total OD on a biolon plate. What happens with the SICA? In some cases, the total growth is reduced. So if you see those smaller circles, the total growth or niche width has been reduced. Yet ultimately we determined it didn't matter. What we care about is how big is that red area in absolute terms when the SICA is present. The total growth that you can accomplish when you have that SICA that is not overlapping with a competitor. The question is basically, can a SICA help me out? And so trying to think about this, weapons, are they weapons? Well, maybe antibiotics are multifunctional compounds. And if an antibiotic, again, using these naturally occurring populations, we thought, okay, look at the second hypothesis here. If antibiotics are weapons, there should be selection for resistance, right? If this is a bad thing, evolution is pretty powerful. We should see resistance. But what if antibiotics aren't weapons? What if Julian Davies is right? What if they're predominantly signals? If antibiotics are signals, and in fact might reduce that competitive conflict, maybe you've got a bigger escape from competition, a bigger red growth zone, we shouldn't see antibiotic resistance. And notice I said that different antibiotics did different things as SICA. So how can we actually test this out? Well, there was a wonderful paper, it's an old paper. In 2006, DeCosta et al. Out of Jerry Wright's lab in Canada went all over the world and collected hundreds of streptomyces and just looked at, it was a clinical paper looking at the prevalence of resistance to different antibiotics in natural streptomyces populations. So what we did here, first look at the dark solid black bars. The percent of the escape ratio is greater than 1. Escape ratio greater than 1 means I grow more, that red free space is bigger in absolute terms when a SICA is present than when it's not. So the black bars are what percent of all possible pairwise combinations with that SICA did we see that escape ratios were greater than 1. For chloramphenicol and tetracycline, it was about 20%, not a lot. 80% of the times, SICA didn't do anything good. But for strep, RIF, and vancomycin, look at 40%, almost half of the cases. SICA's actually enhanced your ability to coexist. You did better in absolute growth when a SICA was present. So then we said, okay, so this idea, let's look at what DeCosta et al. Found globally in the percent of isolates of streptomyces globally that show resistance to chloramphenicol, tetracycline, strep, RIF, and vanc. And what we found is the two that don't show evidence of having any beneficial coexistence effect, there's lots more resistance than these three, where in fact, there's a fairly substantial abundance of SICAs potentially doing something good. So why be resistant? So it suggests that, of course, that antibiotics, certainly they can be weapons. In sympatric populations, we see strong evidence that there is selection for ability to kill things that compete with you. But not every antibiotic might be playing predominantly a role as a weapon in natural settings. And so when we think about organisms like streptomyces that have dozens of presumptive antibiotic BGCs, teasing apart what roles they play in the sympatric or naturally occurring communities remains a big challenge for us. And so if you look at the top half of this, this is the original coevolutionary model that we were working on. Antibiotics as weapons might force arms race coevolutionary trajectory, but due to their cost, niche differentiation might be favored. But in fact, if antibiotics are signals at low concentrations here on the bottom half, signals might actually be antibiotics as signals might be a significant strategy for de-escalating the antibiotic arms race, and in fact, facilitating the ability to coexist. If we're hanging out together, I say, don't eat the Nutter Butters, and you say, don't eat the Oreos. We just put a little bit of antibiotic out. We invest much less. Does this actually end up making us, it facilitates interactions, but of a different outcome, right? And at present, we're exploring this in much more detail among populations that come from different nutrient habitats and plant hosts to try to understand how finely we can slice this idea of antibiotics working as signals that actually facilitate rather than eliminate coexistence. And I want to finish up, and I loved Ophelia's talk this morning. It was a lot of fun because, and all the network structures and images of all different types that I've seen today, everything I've done presented to this point has been pairwise, and averages of pairwise and frequencies of pairwise. But of course, in the real world, microbes are interacting with hundreds of other microbes at the same time. And on the left, each number is an individual isolate. That maps one of our communities of 10 streptomyces with an arrow indicating which isolate can inhibit which other isolate. We're mapping some of these for seeker responses as well, which is pretty exciting. On the right, we have fusarium isolates on the left-hand column and streptomyces on the right-hand column. All of these different things are happening at once. How do we capture this? And more importantly, if we think about antibiotics as weapons and signals and potentially playing different roles for different partner organisms, this really highlights the certainly significant role of higher order interactions when one species alters the interaction between others in terms of secondary metabolites within soil microbiomes. Now, signals, as you know, antibiotics are often signal dependent. Production of antibiotics are signal dependent, but antibiotics themselves can also be signals that incite antibiotic production. In mapping some of these communities, here we have 3 communities on the top. Those are the mapping of signaling interaction, excuse me, of inhibitory interactions. Super complicated. It's a jungle. But then we went further, and in the lower set of diagrams, we mapped enhancement and suppression of antibiotic production. And that's also extraordinarily complicated in these communities. But you begin to see just immediately the potential for higher order interactions. And I'm just highlighting one simple one. 18 and 17 on the top, they don't inhibit each other. They coexist happily. But on the bottom, 18 increases antibiotic production by 17. Go back to the top, 17 kills 12, which kills 18. Enemy of my enemy is my friend. One of the classic predicted higher order interactions that might be occurring in communities and is selectively enriched. And so we've gone a little bit further with this to try to map out the ways that SICA, sub-inhibitory concentrations of the antibiotics, may foundationally rewire communities in ways that lead to higher order interactions. So what we've done here, we took a community of 10 sympatric streptomyces, and it was that center community of 10 from the previous slide. And what we did is we determined SICAs of chloramphenicol, RIF, et cetera, et cetera. Again, these are 6 antibiotics in this case, all of which are produced by streptomyces, and some of which are produced by isolates in this collection. And what we did in this case was looked at nutrient use phenotypes, again, using a biolog plate, and we mapped competitive interactions. That is, if there's an arrow from one isolate to another, that means that isolate has 75% or more niche overlap with the isolate at the end of the arrow. And the intensity of niche overlap here is colored from yellow to dark blue. Blue is more intense. The more intense the color, the greater the competitive interaction. And what you'll see immediately is that antibiotics at sub-inhibitory concentrations foundationally shift resource competition in these communities. You look, for example, at D. D's a king in control, right? That guy's crushing everybody. But you go over to rifampicin, not the same story anymore. Similarly, you look at I. I can go from being, if you look with streptomycin, I stinks. But in many cases, for instance, with streptothricin, it's largely on its own. So you see here that the secondary metabolic activity, these antibiotic biosynthetic pathways, when those genes are expressed at small levels or concentrations of antibiotics are sub-inhibitory, they've foundationally changed the strength of primary metabolic interactions or the pattern of primary metabolic interactions within that community. So how do we possibly go from here to management or even understanding? And I want to finish up. With one of our JGI projects. One thing we found with JGI that's been a thrill and a challenge When you do a project with JGI, you get a fire hose of data, right? And then what you have to do is you have to figure out how you can get the analytical bandwidth in your team to do it. I see you. Yes, yes, yes. And we've been slow to the bolts, but with this project right now, we have had the thrill of we got a NSF Signals of the Soil grant, which has allowed us to finally get analytical firepower. Co-collaborators, Mike Smansky at the University of Minnesota, Will Harcombe at the University of Minnesota. I could not have better collaborators. Postdocs, graduate students all listed there, amazing. We're also collaborating with the University of Manchester. It was a joint UKRI NSF program. And then of course the data that we've been able to generate, or the JGI has wonderfully generated for us. This project, what we did is we took those 10 streptomyces that I've leaned in hard here, and we paired them in 22 sets of 2. And then we had sets of 4 that we co-plated, and then sets of 8, and then sets of 10. And we specifically then have been studying how does the expression of presumptive antibiotic VGCs vary depending upon who you're with and the complexity of those communities. I'm going to just show you some of the wonderful work. This is Stephen Heinz, is a PhD student. He's defending in October and has done an absolutely splendid job in putting the data together. This is just a simple subset or simple summary, but it looks complicated, so I'll explain it briefly. This is 22 possible, or this is 22 pairwise combinations. And here we have 8 isolates that are represented here in different colors, 321.1, et cetera. They're coded by the colors. There's 31 different characterized VGCs. Each one we have a little histogram here. The bars on the histogram is the expression of that antibiotic biosynthetic pathway when the individual strain is in axenic culture, when it's alone. And then what we have, there's red and blue lines. And if you look here, red lines are overexpression of that gene in combination. Blue is underexpression. I'm just going to point out 2 interesting things. There's lots here that we're unpacking, but we've highlighted here 2 different VGCs. One on the left here, this number 13, JBIR 126. It is always turned down or off with every single organism that it's paired with. In contrast, streptothricin is always turned up. It's always turned on in every single organism that it's paired with. We think that these actually, we have some really fun hypotheses about why, but we recognize that looking at the way that these patterns of up and down regulation go, these are broad community shaping chemicals, and who you're with absolutely matters, which doesn't surprise anyone in this room, but recognizing the multifunctionality of these compounds, I think we have a wonderful challenge in front of us. So at the end of the day, what's been fun today is species interactions are where it says species interactions mold the functional capacity of these soil microbiomes through 2 important pathways. One is through selection, and the other is through mediating expression. So selection and expression, 2 different things. Understanding the way these primary and secondary metabolic interactions generate a particular functional outcome in the microbiome is going to be key for us to developing effective management. And again, I want to lean into this again, distinguishing the evolutionary selective effects of the interaction versus the ecological effects, the real-time gene expression. It's really critical for management. Those are different management targets. And finally, this is just last 3 things I want to say. It's been a great day today. I love coming to this meeting. I love people studying co-evolving, coexisting populations, realizing that nature has done wonderful experiments for us. Finding ways to lean into these to dissect roles of evolution and ecology becomes interesting and important for us to make progress. I also have to say it's a golden era for microbial ecologists in terms of development of new ways of thinking. Today, my mind has been blown multiple times. We aren't just looking at cool phenomena anymore. We're trying to develop the frameworks and the hypotheses and the conceptual systems to be predictive. And that's what management's going to take. And at the end of the day, I can't thank JGI enough for really being a force in driving complex analytics to help us get better at teasing apart all these layers of data that characterize how communities work. finally, I want to thank the funding sources and the people in my lab. I realized looking at other people's acknowledgements, this is way messy, but there you have it. And I'd be happy to answer any questions if you have them.
Wow, that was just terrific. That really just what a great way to end the day. It's a talk that pulled everything together from the day. All right, I'm gonna take one question because I'm feeling generous. And Aaron, your hands up.
Thanks for a great talk. I may be missing something, but does it matter what medium you choose when you're defining the subinhibitory concentration? And does that enhance like what you can take away from then testing it on different carbon sources?
So there's two quick answers to that question. First of all, the biolog, it's just a single carbon substrate, and we can't change the nutrient concentration. But that study, the old study, we've repeated that, and our graduate student Molly Cruz is doing fabulous work. She's got a collection of isolates from long-term prairie and a second collection from long-term prairie that was amended every year with NPK, so it's high and low nutrient. And what she's done is she's been evaluating SICAs for all of those isolates on high and low nutrient medium. And so it's an interaction. What's the long-term selective effect versus what's the ecological effect of what the medium I live in now does? So she'll have that answer for you. She's going to defend next spring, so look for it.
Thank you.